related to autism. So early life stress is a major contributor to many developmental neuropsychiatric disorders. Kids, okay. So, if kids go through abuse or neglect or household dysfunction, and they may be, impacted physically, emotionally, sexually, and that results in mental health problems, and that later on could give them developmental psychiatric disorders. So I was surprised to find from CDC that actually many of us, the prevalence of the adverse childhood experience is pretty prevalent. And here you go, okay. Is it not moving? Why? Okay. All right, so the statistic shows like about 61% of adults may have at least one of those ACEs, that stands for adverse childhood experiences, and 16% of the adults could experience 4 or more types of ACEs. And if we find ways to intervene at some early stage, we could reduce up to 44% of the depression in the adults. So it's a big number, and the kind of impact of early life stress could be really significant for people's mental health later in life. Okay, trying to move this. Okay, all right. So many of the psychiatric diseases can have a neurodevelopmental origin. As you can see, many, autism, ADHD, intellectual disability, so on and so forth. Many of them can have a neurodevelopmental origin. Okay. So there are many neurodevelopmental disorders you've heard plenty about. Depression, psychosis, anxiety, and including motor function problems and cognitive problems can all be originated from neurodevelopmental issues. And a lot of study has looked at the genome-wide association and determined many of the genes that's related to these diseases. So many of these genes you look at, and they typically have something to do with the synaptic structure of the neurons. But what we have to recognize actually is that many of these mutations contribute to a small percentage of the population that has the problem. And many kids don't have any of these mutations and still develop all of these problems. So that takes us to think about the environmental factors that, like my kids don't have these mutations, but they can still develop neurodevelopmental disorders. And so we can look at environmental factors like early life stress, early life immune activation are typically linked with the development of these problems as well. And one of the common mechanisms that have been thought about is neuroinflammation. So when you do have early life stress or early life immune activation, the brain is exhibiting some type of neuroinflammation. And one of the molecules that has been documented in the literature quite a bit is this interleukin-1, which is a master regulator of the immune response, and that seems to mediate much of these developmental issues. Now, what I wanted to intrigue you about is that the classic theory actually do not quite address the problems related to most of the kids that has these problems. So the classic theory was pretty much borrowed from immunology, that when you think about inflammation, you think about a frank inflammation, that if you have cytokines like IL-1 in the brain, you trigger leukocyte infiltration, you activate microglial astrocytes that produce more cytokines, produce more reactive oxygen species, creates some kind of damage to neurons. So it was a damaged idea. But most of the kids don't necessarily have this kind of frank inflammation in their brain. So there may be other ways for the cytokines to act in the brain to cause problems. So this leads us, my lab has been looking at IL-1 signaling in the brain quite a bit to try to understand more of a fuller picture of how IL-1 acts in the brain. What's the difference between the IL-1 activity in the brain versus the periphery? And when you look at that, some of the more recent studies have shown that the IL-1 activity can influence many of these processes that seem to be directly impact neurons. For example, the neuroprogenitor cell, the amplification, the neurogenesis part, the neuronal migration, or synaptic pruning, or the phagocytosis of dead neurons. Many of these processes seems to be directly related to neurons. So the question is that do we have this IL-1 signaling through its receptor? IL-1 has one receptor. It's called type 1 receptor for IL-1. Do they bind and can they act directly on neurons? And that was a question that has not been answered clearly in the literature. So in order to get that question, one of the problems, the IL-1 receptor expression on neurons is very low. It's very difficult to see it in the literature. It has been difficult to locate the mRNA expression of IL-1 receptor. It's even harder to locate the receptor, IL-1 receptor. So my former graduate student, Xiao Yu, has done this work and created this mouse we call IL-1 receptor restore, which is we put an interference sequence in the intron 9 of this IL-1 receptor allele and flanked it with the LOXP sites, so later on we can cut it off. So basically, this allele is basically a knockout, so it doesn't function. But later on, you can take it out, and you take it out with a Cre-recombinance. You can do it in a cell type specific manner, and that allows you to control what type of IL-1 receptor you restore in the tissue. And on top of that, because it's difficult to see it, so we put a 3HA tag behind the IL-1 receptor protein, and we put iris TT tomato behind here to visualize IL-1 receptor expression, mRNA expression. So after we did that, we were able to see that the IL-1 receptor expression very vividly in the brain. We see many of these non-neuronal cell types. That's pretty understandable. They're on endothelial cells, astrocytes, choroid plexus cells, and ependymal cells. But we did find them on neurons. And they're very specific. Look at the hippocampus. You see the IL-1 receptor is located almost exclusively on the dentate gyrus, not on the very hard to find it on CA3, CA1 part of the neuron. So it's a granule cell. And later on, we were able to determine there are only glutamatergic neurons in here. So they are very selective in neural circuitry in the brain. And then my postdoc, Danny Nemeth, further mapped neuronal IL-1 receptor in the brain. It turns out it's in the dentate gyrus. The most prominent expression of those neuronal IL-1 receptors in the dentate gyrus and in the dorsal raphe, and then some of the cortical sensory neurons. So they are very specifically located in selective circuitry of the brain. And here, with the help of the HA tag, you can see this is nice. You can see in the dentate, you can see the dentate granular neurons. If you label HA, HA is tagged onto the IL-1 receptor protein. You can see they are right on those granular cell neuron protein. And they are on the surface. If you go high power, you can see they are actually membrane located right on the surface of these neurons. And you can see, of course, there's non-neuronal cell IL-1 receptor as well. And what you use this kind of model, and then with this design, we can specifically restore this receptor only on neurons, only on astrocytes, only on endothelial cells, and so on and so forth. We have several papers, I'm just going through this quickly, that this paper shows that if we restore it on the non-neuronal cells, and you get the classical reaction. Basically, the IL-1 act on those non-neuronal cells, like endothelial cells and astrocytes, produce reactive oxygen species, and that can damage neural structure. So that's fine. That's going with the classic idea. But what is new is that if you restore IL-1 receptor only on neurons, they actually seem to drive social behavior deficits after stress. So the neuronal IL-1 receptor is doing something very different from the classical IL-1 receptor on the non-neuronal cells. In fact, if you restore IL-1 receptor only on neurons, you can put as much IL-1 in the brain as you want. You will never induce inflammation. Basically, there's no microglia activation, there's no leukocyte infiltration. The neuronal IL-1 receptor won't do it. It does something else. So that's something that we were interested in. And this leads us to the work of my graduate student, a neuron, Kochek, and she's changed her name recently. She has begun to look at development and thinking, what is this neuronal IL-1 receptor that seems to have a special role in neural structure and function. So she looked at the animals at the early stage, very early on. She mapped out all the neuronal IL-1 receptor in the brain throughout development. And to her surprise, these neuronal IL-1 receptor are expressed in a sort of developmentally dependent manner. And she saw some, like in the adult, we pretty much just see those few circuitries. But we did find the neuronal IL-1 receptor expression during development in places we've never seen in the adult. So they must come and go during development. And there's some neuronal IL-1 receptor expressing this progressive pattern. They express more and more over time. And the other neuronal IL-1 receptor is expressing certain developmental windows. There's an early developmental window, transiently there's the expression, and then there's late transient developmental window. Certain neuronal IL-1 receptor gets expressed. For the serotonergic neurons, they seem to persistently express throughout life, from early, probably in the embryonic stage, all the way through life. I'll give you some examples. It's kind of interesting. If you look at the dentate, we've looked at, see on P7, on postnatal day 7, you only see this one layer of cells. So the sort of the most mature layer of the cells in the dentate that would express IL-1 receptor. By a week later, you can see the layer has fattened by 3 layers, and then by P20, you got a lot more neuronal IL-1 receptor expression in the dentate. So they're progressively adding more and more. By the time you get to adult, all the granule cells are expressing. So there's this pattern. Then if you look at the, over here, if you look at the thalamic area, so this is the VPL, which is relating your tactile sensory information to the cortex in the thalamus. And you can see actually on the P0 early time, you don't have much, but on day 7, the neuronal IL-1 receptor just pop. And then by day 14, it goes away. So it's the time that this is known for this area to have a lot of reorganization when the sensory input come in to this area, and you have this kind of pattern. And then if you go to the visual cortex, you can see it's a delayed pattern. By day 7, you see all these endothelial cells are expressing IL-1 receptor. This is non-neuronal IL-1 receptor expression. But day 14, the neuronal IL-1 receptor just pop. And this sort of coincides with the mouse that they open their eyes on day 10 and day 12. They start to have reorganization of the visual cortex around this time, and later on they are diminished. So there's this transient developmental window that coincided with the neuronal IL-1 receptor expression. So that makes us think that maybe there's something really important for the IL-1 receptor to function in this developmentally related manner. But the dorsal raphe neuron, the serotonergic neurons, are persistently expressed throughout life and until adult. So what we know is then she did something the neuron did something trying to see what if we change these, can we alter these neuronal IL-1 receptor expression during development? So she put on some maternal separation stress. okay? So just simply separate the pops from the mom for an hour, that's stressful. And what you can see is this increased IL-1 receptor expression in the sensory cortex. It shows a patch. And what is interesting is this patch is coinciding with the patch that will show up after the stress in the cortex in the new N expression. A new N can be seen as one of the, sort of a neuronal maturation marker. So it seems like the signaling of the IL-1 receptor, neuronal IL-1 receptor, is coinciding with these, with the neurons in terms of its maturation. And because also in the literature it's been suggested when you have stress, maybe you have more IL-1 produced. So she said, well, maybe we can try to give a little IL-1. Do we get the same kind of induction? What is interesting is when she gave IL-1, she gave pop IL-1 here, and this is when the blood-brain barrier during this time is not very well formed. IL-1 can still get into the brain. And what she was able to see is that there is an induction of the neuronal IL-1 receptor, but the new M patch is not there. So maybe the neuronal IL-1 receptor can be induced by IL-1, but maybe there's still a sensory input to, sort of synergize with the effect to really changing the developmental trajectory of the system. So then she wanted to find out whether there are any kind of behavior consequences. And what she did is she did this maternal separation for 14 days, and she would look at the behavior 60 days later, and using the same test that our previous reviewer, the speaker had nicely demonstrated using the social interaction and social recognition test. So the social, again, over here is an empty cage versus a animal, and in this social recognition test, it's an empty cage versus a familiar mouse and a novel mouse. So to test whether, does the stress make a difference, and does the neuronal IL-1 receptor make a difference? So that was her test. And it turns out, just show this. It turns out that if you see the social interaction, after the stress, you can see the social interaction is reduced, but in the, let me see. Yeah, so this is control compared to it goes to the animal versus empty cage. This is animal versus empty cage. There's a reduction, but this is not as significantly, it's less significant compared to this. But actually, if you look at a heat map, it's much more dramatic, the difference between the control versus post-stress response, that actually investigate the other chamber. Sort of after the stress is much more less discriminative versus the without the stress, the animal investigated animals much more than the chamber with the empty cage. Now, in this case, when you want to test the animal able to recognize the familiar mouse versus the novel mouse, you can see the control is doing well, but after the stress, it's almost like it's a little reversed. So it's going to, there's no difference. It doesn't know the difference between the familiar versus the novel animal. Now, what is interesting is with our model, you can put you can restore IL-1 receptor on every cell. So we call it GR global restore, and you get this kind of phenomenon. But if you only restore IL-1 receptor on non-neuronal cells, remember in the brain it's either non-neuronal cells or neuronal cells. So you use this Tie2 Cree to restore IL-1 receptor on non-neuronal cells, and this phenomenon goes away. And actually, if you social recognition after the stress, the animals are, the discrimination index almost flipped. It goes to the empty, goes to let's see, it goes to the wrong animal more. But in the IL-1, if you restore IL-1 receptor in the non-neuronal cells, this phenomenon once again is gone. So this, I think this is still preliminary. We're trying to reproduce this and then figure out the mechanism. And I have My postdoc is working on the mechanism how the neuronal IL-1 signals, receptor signals, it signals in an entirely different way compared to the classical, IL-1 receptor that's on macrophage. So let me just summarize, and that what we found is the IL-1 receptor is expressed in multiple cell types in the brain, in the dentate granule cells and the dorsal raphe neurons, that's in the adult. And the neuronal IL-1 receptor is expressed dynamically during development, some only expressed in specific developmental windows. Maternal separation stress and prenatal IL-1 challenge could increase neuronal IL-1 receptor expression. Maternal separation can cause social behavior deficits later in life. The stress-induced, these changes may require neuronal IL-1 receptor. And it seems like the nervous system is hijacking the neuronal IL-1 receptor, but doing something very different from the classical way of signaling in the immune system. It's not triggering any inflammation, but it's still sufficient to change developmental trajectories, and that may contribute to the development of behavior issues later, the change of neurodevelopment. Neural structure, misconfigured neural structure. So let me give you the acknowledgement, and the people contributed to this work in my lab. This work is critically done by my graduate student, Neuron. She's got a poster here. There's more detail. You can go to her poster, and then my postdoc, Danny Nemeth and Matt Schreier, and my lab manager, Sam McGovern, and my former graduate student, Xiaoli Liu, my graduate student, Maria Neuron, and this is an undergrad in my lab, Numana, and we also collaborate heavily with Dr. Randy Blakely's lab, and my former postdoc, who's now a lab chief in China. We collaborate with him on there's some electrophysiology he did I don't have time to talk about today on the neuronal IL-1 receptor. And my finding sources from NIH. So I hope I kept it short. Thank you very much.